You know about the dihybrid cross, already you have done in your class. Okay, so if the situation is given, let's say capital A, capital B, capital B. So why this is a dihybrid cross? Because two gene is mentioned. This is gene number 1, this is gene number 2. Voice clear? So these are the two gene. Okay, gene number 1 and gene number 2. So if 3 gene is mentioned, then it will be a trihybrid cross. 4 gene is mentioned, that it will be a tetrahybrid cross. So it based on the number of gene. Like yesterday, whatever the point we have discussed, height of the plant, that is only one character. It might be tall, it might be dwarf, but these are contrasting character. We are not talking about in a single plant tall and dwarf present. That is not possible. Getting my point or not? One plant will be tall and dwarf both. My height is tall and dwarf both. That is so not possible. One character it will reflect. So if the situation is this, how the law independent assortment will be applicable? See, this A with this B, so A and B, comma, same A with the another B, this A with this B, these are gamete formation. Remember that. Whatever I am forming the gamete. Please remember the step, always follow the arrow. Without arrow, don't do this. Getting my point or not? Without arrow, don't solve any problem. So this is the homozygous situation. Homozygous A, homozygous B gene. Both gene are in homozygous situation. Clear or not? But if the situation is given, that example that I give you yesterday, capital A small a, capital B small b, then how we will solve? Same process, same arrow we will do. Capital A, capital B, comma, same with the capital A small b. So capital A small b, comma. Now small a with capital B, small a with capital B. Yesterday also I discussed why small a will come first before the capital B, because the first gene is A. First gene is A. So always this is room number 4, that is room number 5. So always the room number 4 will come first, no matter it's a teacher or student, because their position is first, then that room will come. Getting my point or not? Okay. Next. And the last one, this small a with this small b. So these are the 4 gametes. So what are the basic difference here? What are the basic difference? How many gamete form? How many gamete? 4. How many gamete here? 4. In monohybrid cross, let's say this. So how many gamete will form here? 2. 2 by 2. No? That is in monohybrid cross. So in monohybrid cross, how many gamete form? 2. In dihybrid cross, how many gamete form? So without solving the trihybrid cross, how many gamete will form? 8. 8. So in tetrahybrid cross, how many gamete will form? 16. So how you are going? 2 to the power n number. Understand that these are the things which is not written. This is the kind of chapter where you have to understand the basic. Question can be asked in various grounds. All problem detail solving is not required if you understand the particular topic. Like the way I discuss. General student, If such question will be asked that how many gamete will form, every one of you will be engaged to solve. Not required. If the number of gamete they are asking that how many gamete will be formed, simple. So write down. Total number of gamete. If the question asking how many gamete, total number, formula is 2 to the power n number, where n equals to types of cross. Type of cross. So example, if the cross is monohybrid cross, so 2 to the power 1, answer is 2. 2 gamete will come. Getting my point or not? If that is a dihybrid cross, then 2 to the power 2 is equal to 4 will come. Like that came here. If that is a trihybrid cross, then 2 to the power 3 is equal to 8 gamete will form. Same, if the tetrahybrid cross will be there, 2 to the power 4 is equal to 16 gamete will form. Clear or not? So here without solving, understand that, without solving the problem we can easily answer by checking how many gene is present. If the number of gene, because 1 gene monohybrid, 2 gene dihybrid, 3 gene trihybrid. So the type of cross or the gene is given. Based on that, we can easily answer this kind of question. Clear or not? Okay. Chalo. Second problem. If you carefully check this. Now my next question. Chalo, total to 4 gamete will form in dihybrid cross. Total number of gamete number is 4 in dihybrid cross. But here, how many gamete genetically differ? How many gametes are genetically different? How many? No. All are same. All are same type. They are not genetically differ. They are same type. Is it or not? But if you check here, all gametes are genetically different. One is capital A, capital B. Capital A, capital B combination in other of the 3 here, it's not there. Getting my point or not? That is genetically different. So how many gamete is genetically different? 4. How many here? None of them. Only one type produce. Out of the 4, only one type is here. But out of the 4, all the 4 types are different. Getting my point or not? So, write down. This is the second part. Here too, we calculate total number of gamete. Now, total number of genetically different gamete. Genetically different gamete. Formula same 2 to the power n number. Formula same 2 to the power n number. Remember this. These formula not given in the book. These are simple strategies. 2 to the power n number. Clear? So where n is equal to number of heterozygous gene. Number of heterozygous gene. How many heterozygous gene present? Tell me. How many heterozygous gene present? None of them. None of them. Clear? How many heterozygous gene present? 2. A is also heterozygous. B is also heterozygous. Is it or not? So A is also heterozygous. B is also heterozygous. So if 2 is heterozygous, so how many gamete will be genetically differ? 2 to the power 2 is equal to 4. Getting my point or not? So write down, with the help of this, write down one problem. If the genotype capital I small I capital H small h capital G capital G capital K produce p number of genetically different sperm, p number of genetically different sperm, p number of genetically different sperm, and the genotype capital A small a capital B capital C small c capital D small d produce q number of genetically different ovum, find out the value of p and q, how many gene is here? 1 2 3 and 4. Here how much gamete can form? 2 to the power 4 is equal to 16 gamete can form. 16 gamete can form. But question asking that genetically different sperm, p number of genetically different sperm. Asking the number, how many will be genetically different? This is one gene where it's heterozygous. This is second gene where it is heterozygous. So 2 to the power 2 is equal to 4 will be answer. 4 gamete will be genetically different out of the 16 gamete. Bas, answer done. If you go with the traditional process, then you have to do this, and it will take huge time. Getting my point or not? Because they are not asking what are the gamete. Then we have to solve. That part I am coming. Here they are asking how many, number. So number, obviously it's a number game. Now here this is heterozygous, this is heterozygous, this is heterozygous. So total 3 heterozygous. So q is equal to 2 to the power 3 is equal to 8. Total number of gametes same. Total number of gamete will be 16, but out of the 16, 8 different types of gamete will be formed. Bas. Parental generation given as a capital T, capital T, capital P, capital P. Be sure about your handwriting. Cross with small t, small p, small p. So this character is a tall and purple. This character is dwarf and white. Okay. Now, so how the gamete will form? Gamete formation, already I told you that law of independent assortment will be applicable here. So all the gamete will be total 4 gamete, but these 4 will be this. Cross with small t, small p. Same 4 by 4. NCERT have shown only one side, only one. Please do not follow that process. So here 4, here also 4. So 4 by 4 gamete. So there will be 4 by 4. That means 5 by 5 checkerboard we will take. Now capital T, capital P, small t, small p. Be sure about that, which one you are writing as a capital letter, which one you are writing as a small letter. Okay. So among these, so how you will fill up the box? This is the row wise and this is the column wise. Now first row, first column, I have capital T, capital P, small t, small p. How the arrangement I will write? Among them, which gene is given first? T. Height gene is given first. So among the height gene, capital T will come first or small t will come first? Capital T. Then the small t will come. Next capital P, small p. And remember that all of the boxes will be same. Clear or not? All the box. How many box? 4 by 4 checkerboard we have formed. So 4 4s are 16 will be in the box. So phenotype ratio will be same, 16:0. So what are the character? Tall and purple, but they are heterozygous character. Now next step, F1 cross, and we know F1 cross is called as a selfing. F1 cross is called as a selfing. So capital T, small t, capital P, small p, cross with capital T, small t, capital P, small p. Now again F1 will form F1 gamete. So how the gamete will form? Law of independent assortment. So capital T with capital P, comma. Remember that this is a standard process, and from here tricks will be applicable. The way same I am forming the gamete, please follow the same process. Second, again this capital T, small p. So capital T, small p. Be sure about your handwriting. Third, small t, capital P, small t, capital P. Next, small t, small p. Here also same will come. So I am not writing here. Clear? So F2 generation checkerboard will be, in the F2 generation checkerboard, again 4 by 4 gamete. So 5 by 5 checkerboard we have to form. First is capital T, capital P. Second, capital T, small p. Third, small t, capital P. Fourth, small t, small p. Okay. Please, the way that I am forming gamete, because understand that much of time will not be there in the exam hall to do the dihybrid cross and to solve the problem. At the end, tricks will apply. Only the tricks will be applicable if you follow the same. That means how I have formed, which T I have taken. That's why I have marked with a different color. First this T, then I have distributed both of the P. Then I come to the small t and distribute both of the P. Clear? So here capital T, capital P, capital P. Next, capital T, capital P, capital P, small p. Next, always place your left hand side finger. Understand, here to dihybrid cross we have to deal with 16 box. Trihybrid cross will be there, 8 by 8, 64 box. So if one mistake, 2 days, 3 days will be required to find out. So please place your left finger and check. row and column wise. So capital T, small t, capital P, capital P. Next capital T, small t, capital P, small p. Next capital T, capital T, capital P, small p. Next capital T, small p, small p. Next capital T, capital T, small t, capital P, small p. Capital T, small p, small p. Capital T, small t, capital P, capital P. Capital T, small t, capital P, small p. Small t, capital P, capital P. Small t, capital P, small p. Tell me. See, this is the board, and if you follow this board carefully, if you follow this board very much carefully, understand that where the tricks in the color, in standard Mendelian chart, in standard Mendelian dihybrid cross chart, always suppose any cross come, we will not do this process step. What we will do? We will check the problem and we will remember this. Just we will remember. If you check carefully, first box, all are dominant. Next, single tick. Wherever I am marking the single tick, these are the 9 position, 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 position, tall and purple character are there. Double tick means, see, all are dominant. Single tick means minimum 1 capital T. Second, does not matter, maybe capital, maybe small. Minimum 1 capital P will be there. So that character will reflect tall and purple in our chart. So how many tall and purple? 9. Second position does not matter for us. If capital T, that will also reflect tall. If capital T, small t, that will also reflect tall. Getting my point or not? Okay. Now, in our chart, this green color portion is 3. Total 3 portions are there. These are the area where you can see T need to be dominant or heterozygous, but P need to be small, that is tall and white. How many in our chart? 3. Tall and white within our chart is 3. Red color in our chart, this. Here both the T need to be small, P minimum 1 capital. So here dwarf and purple. How many within our chart? 3. The last that I have written, this one, here all are small. How many within our chart? Dwarf and white? 1. So this is the 9:3:3:1 ratio.